Okay, today we're going to be talking about the microbial mechanisms of pathogenicity, Chapter 15 lecture. The signs and symptoms for a disease can be problematic if they are not unique for a particular pathogen. So this is why they try and make signs and symptoms as specific as possible for a particular type of pathogen. So signs are measurable and objective. Symptoms are subjective, so how a person feels. Whereas signs are like a temperature and heart rate. Those are things we can measure. You need to know these terms for the exam. Nosocomial disease is a disease that is acquired in a hospital setting, and a zoonotic disease is transmitted from animal to human, like COVID-19. You need to know the rest of these terms, though, for the exam. Remember, things that are eukaryotic are harder to kill than things that are not eukaryotic, because we are eukaryotic, and that means there's less difference between us and eukaryotic pathogens. So these are more terms that you guys need to know for the exam. I'm not going to go through them on the video because it's just stuff you can learn by reading through it. So the 5 stages of disease, you need to know these 5 stages of disease, and of course the number of pathogens increases over time, and then once your immune system kicks in, the number of pathogens decreases over time. And I think we all know what acute disease, chronic disease, and latent disease is because we have gone over that previously in class. Koch's postulate, you guys have gone over this in lab, and I've talked about it in lecture as well. Make sure that you understand how it works. So the LD50 is a lethal dose for 50% of the population. This can be for a pathogen or this can be for a drug. So for instance, if the LD50 takes less pathogen to reach 50% of the population to be lethal, that means that pathogen is more lethal to the population. If the drug takes less dose to reach the LD50 than another drug, that means this drug is more lethal than this drug. So you can read through this. This is notes that I wrote down during lecture. You can read through them. The ID50 is the infectious dose for 50% of the population. So how do you infect 50% of the population? So if you look at the ID50 for something like Giardia, well, the ID50 is 1. It takes one organism for 50% of the population to get infected by it. So if you have one Giardia in your system, 50% of the time you will get the Giardia disease. And so you can see here that for rotavirus you need 10 to 100 viral particles. It sounds like COVID-19 is shaping up to be about 1,000 for the ID50. Portal of entry, how do pathogens get in? Remember that needle is one way that pathogens get in. Biofilm. So on a catheter, the UTI is the most common nosocomial infection, and those pathogens are becoming antibiotic resistant. Remember that the biofilm allows for the survival of a pathogen on a hard surface for longer periods of time than it would be able to if it did not have that biofilm. And they become antibiotic resistant because they are able to work together in conjunction to diffuse the concentration of antibiotics over a biofilm. So invasion, so H. pylori, it's known to cause ulcers. What it does is it will release urease, an exoenzyme, which will neutralize the stomach acid and cause the breakdown of mucin, which is the mucus that your stomach makes to protect those epithelial cells. So once it breaks down that mucin, it is able to then penetrate those epithelial cells in the stomach. So urease is an exoenzyme. So it's a virulence factor because it allows it to spread further and proliferate. So pathogens capable of crossing the placental barrier, these are the ones that are able to cross the placental barrier. And so these are the ones you have to worry about if you have a pregnant patient, because it can cause damage to the fetus. And so, like for instance, syphilis is one that you have to worry about if you have a pregnant patient, because then the fetus could be exposed to that particular pathogen. Know these for the exam. Portals of exit, you'll notice that a lot of these portals of exit are the same as the portal of entrance. So how pathogens get in tends to be the same way that they get out. Cellular damage, swelling, is called edema, and that is caused by inflammation. And what it's used for is to restrain a pathogen in an area of the body. So this goes over the edema. So an injury, disease causes, we give medications and that can cause edema, and we get exercise can also cause edema, but these all are causing some sort of damage, and that's some sort of cellular damage. So a white blood cell, like that is macrophage that can do phagocytosis, like a dendrite, macrophage, neutrophil, or even a B cell. When it releases cellular debris that become of a pathogen that it's released, that then upregulates the immune system to recruit other white blood cells there that will then release cytokines or recruit complement proteins to that area that cause inflammation. So when there's damage done, cellular damage done, you will also have the cleaning out, phagocytosis of these damaged cells, and that will then release cellular debris as well. So that then causes upregulation of this phagocyte to release cytokines, and these also recruit other white blood cells that will release more cytokines and recruit complement as well, which will then also upregulate inflammation. So when you have the release of these cytokines, that causes the blood vessels to become permeable, so the liquid comes out, which causes a swelling. And the liquid comes out that you can allow more white blood cells to come in through that blood vessel. So remember, the lymphatic system dumps into the blood. The blood can only have things come out into the tissue, and then those things can go back into the lymphatic system. So it's a circular process. So virulence factors, for instance, protein F, that sticks out from Streptococcus pyogenes, and that allows for attachment to the respiratory epithelial cells. These are just different virulence factors that stick out from these particular pathogens that allow them to adhere to these types of cells. And this tells you what type of adherence it is. So for instance, gonorrhea uses a pili to attach to the urethral cells. Enterotoxigenic E. coli uses fimbriae to attach to the intestinal epithelial cells. And these are all protein structures. And remember, they come from a gene because we have DNA that becomes RNA that goes to protein. So these pathogens all have genes for these adhesin proteins. Cellular damage is due to an exoenzyme like hyaluronidase. Hyaluronidase is an exoenzyme that breaks down the soft connective tissue between cells. When you break down the soft connective tissue, that allows bacteria to have access between those cells to go to a new area. So that new area has less competition, more nutrients available for that pathogen, therefore it can then survive better. So here are some different exoenzymes. You need to know these for the exam. DNase, for instance, chews up DNA, and that's important because DNA can be released by dying cells that are undergoing apoptosis due to the presence of a pathogen eating up nutrients or causing damage to those cells, and that DNA that's released is like a gelatinous substance, and it's hard for the pathogen to move through. So if the pathogen releases DNase and it degrades the DNA, it makes it easier to move through